Everybody got the sandwich of their choice, sandwich of their desire. I wasn't going to do any introductions because, of course, Dr. Palefsky needs no introduction. So I was going to try to get off the hook by that way, but then I thought it would be very nice to at least formally introduce him to the audience, because I think one of the key traits of Joel is that he's really very modest about all of his accomplishments. So if I don't tell you some of them, you probably won't. So I think that was my motivation for at least saying a few words. So Dr. Palefsky, as you know, is director of CTR Fellowship Program and is professor of medicine here at UCSF, and is really, I think, emblematic of what we try to do here as faculty. And certainly he's been my inspiration and mentor all these years.
Comics here.
Yeah, exactly. So the three spheres that we try to keep up in the air are clinical care, which you've been learning about, and some creativity. So clinical care, education, and research. So I think really there are a few people who juggle these balls really well in the air, and I think Joel is really, truly one of them. And you will see by the end of the talk what I'm talking about. I think he's really led the world in some of the areas in terms of thinking about human papillomavirus infection and disease in men in particular, and most recently prevention of disease, because I think prevention is really the most lofty goal that we can aspire to as clinicians. Thanks a lot, Joel.
Hey everybody, I'm not showing any gross pictures, so you can be away. If you have any questions, pick up your hand and shout it, okay? So I'm going to talk about some of the things that I do, tell you a little bit about moi, and give you a sense of the range of things that Peter was alluding to that we do, and some of the excitement that we have being on the faculty here at UCSF. And it is certainly, I would say, as I said to some of the pups when we had a meeting a few weeks ago, probably the best thing about being in academia in general, about being at UCSF in particular, is the amazing people you get to spend your life with. Peter is one of them, and he did tell you he called me Papa. Usually when somebody's coming asking for money, and people like Sue Ellen, and you know, it's just an exciting way to spend your life. So, that's not me. Some of you are probably too young to even know who that is. So I put his picture up because I was a Doogie Howser just a few years ago. I graduated from medical school when I was 21. And I never actually had a life. While you guys were out having fun, partying as undergrads, I was saving lives. And you know, it actually has its pluses and minuses. Everybody needs to sow their wild oats. So one of the things I'm not going to tell you about today is how I tried to sow my wild oats after I joined the faculty. But I grew up in Canada. I went to Yale University. Has anybody ever heard of it? Okay, we call it the Harvard of the North. Some people have said to me, is that a four-year school? Best university in Canada, great city. But I trained there from pre-med to elementary school, high school, community college, medical school, internship, residency, postdoctoral fellowship, and then finally I decided it was time for some better weather. So I moved to Stanford, did my infectious disease fellowship at Stanford. And then after I was finished with that, I came up here to join the faculty at UCSF, where I have been ever since. So I'm kind of not typical, and neither is Peter, actually, in the sense that we haven't really done a whole lot of moving. Most people in the course of their academic career do switch jobs or universities quite often, but that's just not been my trajectory. So I spend my life thinking about this guy, HPUs, Has anybody here not heard of HPV? Everybody here probably is young enough that they should have been vaccinated, boys and girls. We'll talk about why soon. Almost everybody. This is a computer rendering of the virion, and what it shows are these pentameric structures, which are composed of the L1 major capsid protein. And the part that you can't see is in the middle here, which is called L2, the minor capsid protein. The reason this is so important is because the major capsid protein forms the basis for the vaccine that some of you have already had. And the vaccine consists of that L1 protein expressed in vitro, which auto-assembles into a three-dimensional structure of each cell. The reason that I care about HPV is, as an infectious disease person, it is the most common sexually transmitted agent. About 85% of everybody who's sexually active will get a genital HPV threat at some point in time during their lives. So most of you have it, or had it, or will have it. And that in and of itself is not a reason for alarm. What is a reason for alarm is the diseases that HPV causes. One of the most common ones is genital warts. They don't kill people, but people really don't like them. What can kill people is this stuff here at the more severe end of the spectrum, which we call high-grade squamous intra, meaning within, the epithelial lesion, the HSIL. Here's the basement membrane of the skin. And as long as the cells stay above the basement membrane, we don't care much about it. But what we do care about is when those cells cross the basement membrane into the stroma. That is, by definition, what? What disease is that? Cancer. Right? That is a serious problem because if you let it go unchecked, these spread locally into the pelvis, they block vital organs, or they metastasize the lungs, the liver, and they kill people. And I'm going to show you exactly how many people HPV actually kills. I'm guessing that none of you know anybody who's died of cervical cancer. Is that okay? You know one? Okay, okay, sorry. Most of you probably don't even know anybody who's had it. It's not a very common disease here, but globally it is a major public health problem. And the reason for that relates to prevention efforts, or lack thereof, that in most of those countries where the world's women live, there's no organized screening program to prevent cervical cancer. So that's where most of the cases occur, and that's why most of those women die of what is essentially a preventable disease at this point. It also has important lessons for us in the other disease that I'm very interested in, which is anal cancer, also caused by HPV. It's the same disease, but in the anal canal. And we're kind of like decades behind because at the moment there are no screening programs for anal cancer anywhere, not only in developing countries, but here. We'll talk about that. So here's that skin again, and HPV actually likes to establish itself in the basal layer. That's pretty crafty on the part of the virus because this is basically an immortal cell layer. Your skin is always turning over, right? You're kind of like snakes. You're shedding all the time. It takes about a month to go from here as the cells mature to the top, where they essentially die and fall off. But the virus, by establishing itself here, basically divides, and when the cells divide, so it's pretty much always there. So we actually think that HPV is more like herpes than what you'll read about in the literature. It probably never really goes away. Probably your bodies control it. The vast majority of you, as long as you remain healthy, will not have any problems from it. But in a few people, that dormant virus can reactivate later in life if they become immunocompromised. To get down there, you have to go through all these layers of keratinocytes, including keratin. And the way that we think that happens in most cases is actually a tear or a breach in the skin surface. The way that that happens is mechanical. If you're having vaginal intercourse or anal intercourse, you can imagine it's not hard to have micro tears in the lining. And other things like other sexually transmitted infections can do this, like chlamydia, like gonorrhea. And So the skin surfaces that are susceptible to those other STIs are particularly susceptible to HPV as well. So this shows what happens over time in healthy young women. This is a study, it's one of the classic studies in the HPV world from the Portland Kaiser cohort study. This is 5-year age groups of women, and this is rates per 100,000, so this is a log scale. Here's HPV at the top, far more common than low-grade disease, high-grade disease, and cancer. You'll see that the peak prevalence is in the youngest women, late teens, early 20s. Most women get their HPV within their first 2 or 3 sexual partners. And then you see an age-related decline in the detection of HPV. Remember, it's not going away, but it's becoming controlled, and probably now is below the level of detection of currently available tests. So most women become HPV negative by the age of 30. Now, when you guys reach that age, you'll have a pap smear and an HPV test. If that HPV test is positive, then you'll probably be sent for the next step in the evaluation, which is a colposcopy. To take a look at the lining of the cervix and take biopsies to decide what to do. We do not do HPV testing in people under the age of 30 because it is so common and because most of them are going to be inconsequential. So over the age of 30, HPV testing is growing in importance as a screening method to prevent cervical cancer. Again, the idea is you get an abnormal test, be it a cytology, pap smear, HPV test, colposcopy is where you look at the cervix under magnification to look for the high-grade or low-grade changes. If you find the high-grade changes, we want to remove them to prevent cancer. If it's low-grade, we kind of don't care too much. Now this is the cancer rate. So this is pretty low. In this country, it's about 8 to 10 for 100,000 women per year. That is still leads to about 7,000 or 8,000 cases per year of cervical cancer in the U.S. and several thousand deaths. And you may ask yourself then, if we're spending billions of dollars, which we do, on prevention, cervical cancer, pap smears, etc., why are thousands of women still dying every year of cervical cancer? And the primary answer is not because of the failure of the pap smear per se, but the failure of the healthcare delivery system. Because the majority of women who get cervical cancer in this country are of lower socioeconomic status, have not seen the doctor, and have not had those screening visits. And that's why it's a global problem, because again, as I said, a lot of women live in countries where there's no routine pap smear screening. So this shows you those rates of cervical cancer. They're highest in the orange and yellow countries, primarily south of the equator, sub-Saharan Africa, overlapping a lot with the HIV bill, unfortunately. But there's a few numbers I want you to pay attention to. So one is, these are the actual age standardized rates. And you'll see that the worst countries are listed as between basically 35.8 and 93 per 100,000. Yeah.
Can HIV cause susceptibility to cervical cancer?
Yes, it can. I'm going to talk about that. And so these numbers actually translated to about 500,000 cases per year worldwide, and 250,000 women die every year of cervical cancer. So it's right up there with HIV. In fact, it's the #1 or #2 cause of cancer-related death in women in the developing world. Now, this is a pretty broad number. In fact, in the worst countries, for instance, India, it's an enormous problem because the denominator is huge. And the incidence is about 60 per 100,000 per year in India. Remember that because I'm going to compare that to what we're seeing for anal cancer in people. So we talked about how certain parts of the epithelium are particularly susceptible to HPV. And that's what's called the transformation zone, where two different kinds of epithelium meet. One is called the squamous epithelium of the outer part of the cervix. This is covered in keratin and all that stuff. And then the inner lining of the canal of the cervix is covered with cells that produce mucus. So that endocervical epithelium, where it meets the exocervical epithelium, is a very thin and active epithelium, a great place for HPV. This is where most of the lesions arise. Turns out that in the anus, there is a very similar area. Here is where the anus, this is the anus here, here's the rectum, colon, and here. Where the anus meets the rectum is a transformation zone very much like in the cervix. And so if you're trying to figure out if somebody's got bad stuff going on up there, the only way to do that is actually to do some tests that we basically stole from the gynecologist, and we do them in the anus. So women have been getting cervical pap smears for years. We do anal paps on at-risk people. Basically what we're doing is we're putting a swab up and then sampling the entire area here, including this area. And if it comes back abnormal, then we do a technique called high-resolution anoscopy, or HRA. Actually uses a colposcope, and then we look for lesions, take the biopsies, and if somebody's got high-grade disease, we treat it to try and prevent anal cancer from developing. And I'll tell you why that's an interesting issue right now in just a few minutes. So this is a paper by this guy, and this was something we worked on together. I actually think it's one of the most interesting papers in literature, not because we did it, but because it is the only, to my knowledge, population-based data on the prevalence of anal HPV infection and disease in a standard, if you will, urban men who have sex with men or any population. So what we did in this study is we got people from a cohort that had been recruited through a technique called random digit dial. Basically picked up the phone, said, hey, are you gay? Would you mind if we put something up your rear end? And no real problems to figure. And so they sampled themselves. We taught them how to do that. Then we brought them into the clinic and we sampled them again. So what we found was basically like a random sample of HIV positive and HIV negative men, MSM, in San Francisco. And to me, and the reassuring thing was the numbers are actually very similar to what we saw in our convenience cohort study. And they confirmed basically what we were seeing. What we were seeing was very, very scary. Here, for example, is HPV infection on the top. You'll see here that amongst the HIV positive men, 88% have anal HPV infection. Some of these subsequent studies basically show 100%. Even nearly 60% of the HIV negative guys have anal HPV infection at that one single sampling. Okay. Even more concerning actually is disease, because again, it's disease that really matters from a mortality standpoint. AIN means anal intraepithelial neoplasia, and 2 or 3 is the same as the high-grade lesion. And what we found was that 43% of the guys who are HIV positive, pulled off the street if you will, have a high-grade lesion at any one time and are at risk for developing cancer. And remember I said cervical cancer is 60 per 100,000 women per year. Right? So here 43%, nearly half, of the guys walking around with HIV infection have high-grade anal disease. And if this was in the cervix, we would want to remove that to prevent cervical cancer. 25% of the HIV negative guys have high-grade lesion. And that's in some ways even more concerning because the denominator is bigger. Right? There are more HIV negative MSM than there are HIV positive, fortunately. So, it's not just a problem of men though, because one of the sort of shocking things that we've learned in our studies is that women actually have more HPV infection in the rear end than in the cervix. Okay? And that is, we found that out initially in the highest risk group of women, principally HIV positive women and high risk HIV negative women. This was a study that was done before we were using a lot of antiretroviral therapy. You can see that the lower the T cell count, the more HPV infection there was. Yellow is anal, green is cervical. And even in the HIV negative people, that was true, more anal than cervical. People said to me, well, of course, these are very high-risk women, they're not typical, but we and others have done a bunch of studies in women who are more normal, or representative, if you will, and the same thing is true. We did a little study in Planned Parenthood. healthy girls coming in for contraception counseling, young women, no history of an abnormal cervical patch. 50% of them had a cervical HPV positive test, 60% had a positive anal test. So this is a much more common phenomenon than any of us originally thought, and we're still not entirely sure how it plays into the whole natural history of HPV, even in the cervix, because there's communication between the two anatomic sites. So here's one of the reasons why we care about anal cancer, and that is that, like oral cancer, how many of you heard the Michael Douglas story? Okay, so there's a lot of attention. Okay. I'm going to try and keep this talk from becoming X-rated, but basically Michael Douglas, famous actor, came out and said he had oral cancer due to HPV, and he got it through oral genital sex. True, probably. And it was a big revelation to everybody but us. But oral cancer due to HPV is becoming more common, and so is anal cancer. Since the 1970s, anal cancer is increasing in men and women by about 3% per year. And actually, since you shouldn't be surprised anymore that it's more common in women than it is in men, given that anal HPV infection is so common. So what's actually happening here is that any woman who is sexually active is probably at risk, so the denominator is huge. But because of amongst men, that risk is more concentrated in men who are sexually active and male, primarily men who have sex with men. So here are what the numbers look like. Here in the US, before we were doing cancer of the cervix was about 40 to 50 per 100,000. Now it's down to about 8 to 10, as I said before. Before the HIV epidemic amongst MSM, the estimate was that anal cancer was found in up to 37 per 100,000. So kind of similar to what we were seeing here. Again, it makes sense because we don't screen routinely. Now, in the HIV era, things are even worse. You might have thought that when we put people on antiretroviral therapy, that's going to make their anal cancer problems less. But in fact, for a variety of reasons, it's the opposite. It's actually gotten worse. Once we started people on antiretroviral therapy, the incidence of anal cancer continued to climb. And these are the most recent numbers. 131 per 100,000 in men who are sexually active. Remember, 60 per 100,000 for the worst countries for cervical cancer. Men who get HIV through proper injection drug use primarily, 46 per 100,000, and women about 30 per 100,000. So these numbers are not okay. And if we saw numbers like this for cervical cancer here, there would be a congressional investigation that would not be acceptable. So now we have a new clinical problem. This is one of the interesting things about academia. You get to work on new stuff. And this is a new disease, and I've been very fortunate in my career to be able to work on a new disease. There aren't that many new diseases, but this is one of them. So what are we going to do about it? The first is let's try and understand the molecular pathogenesis, particularly the interaction between HIV and HPV. Second is what can we do to prevent it? And the obvious answer is vaccination, because the quadrivalent vaccine, also known as Gardasil, the bivalent vaccine, known as Cervarix, contain HPV 16 and 18, and HPV 16 and 18 are the most common causes of anal cancer. So, for the same reason that these vaccines should prevent cervical cancer, or a chunk of them, they should also prevent a chunk of anal cancer. And then, what about the people who are too old for vaccination? Can we do something in the anus that's similar to what we already do in the cervix to identify and remove the high-grade lesion? So, you asked about HIV and HPV, and there's a lot of different possibilities. The most obvious one is suppressed immunity, and that's certainly playing a role. But, you do have to focus a little bit when you're doing laboratory science, and the area that we've been particularly interested in is trying to understand what's happening at the tissue level through the effect of HIV proteins. And basically, the skin's main role in life is to keep out bad things and to keep in good things. And it does that in a lot of ways, but one of the ways is it has a zipper. Okay, one of your layers of your skin have these things called tight junctions, which are really a very tight zipper. They keep things out for the most part. But we started to think that maybe some of these proteins that are produced in HIV-positive people might actually break that zipper. And if that's so, perhaps that allows bad things in, like HPV, more viral infection, and more disease results. Why did we think that? We started off with a clinical observation, which was that when we took normal tongue tissue as a model for epithelium from HIV-positive people and looked at it under the microscope, even though it was clinically normal, it was not at all normal under the microscope. So this, for instance, you see these nice pink, red lines. This is under confocal microscopy, and this shows healthy zippers. This is a normal-looking tongue, clinically normal, from an HIV-positive person. I think you can see that it doesn't look the same. It suggests that there's something different going on in the HIV-positive person. And since HIV is one of the obvious candidates, we asked whether it might actually be HIV proteins doing that. This is, so I'm not going to go into detail about this, but this is what some of the zippers look like. These are the tight junctions. And the key proteins are called gluten and gluten. And what we did was we took tissues from people and we grew them in the lab in three-dimensional culture. And did it in such a way as to keep their three-dimensional tissue integrity. And basically what we did was we exposed normal tissue from HIV-negative people to HIV and asked whether we could see the same change. So here's the control. You see that nice red stuff. This is what happens when you add HIV. You see it's lost. And these are actually HIV particles here in green and proteins. So clearly HIV could do that. But that's not really enough. We need to try and understand at a greater level of detail. So we use this technique called polarized cell culture, where we grow the cells in monolayer in a chamber where you can add things here, and then we measure the integrity of these zippers using a technique called transepithelial resistance, or TER. You're basically measuring electrical current, where the lower the resistance, the more disrupted those zippers are. And you can measure that, and you can also look and see what comes through at the bottom. So what we did was we took those cells and we measured TER, transepithelial resistance. These are what happens when you take cells and just leave them alone. Over a period of 5 days, the TER goes up because they're forming those tight junctions. But if you add TAT, one of the key HIV proteins, you see it going in the other direction. So those zippers are being disrupted. The same thing happens if you add GP120, which is another HIV protein. As another control, you can take a mutant TAT that isn't active, and it does the same thing as the control. See it going up here. And if you inactivate the GP120, it also acts like control. And then if you add TAT and GP120 together, it's kind of synergistic. So that suggested not only that it's HIV, but it's these two proteins, either alone or together, doing that. So then the next question is, does that have any clinical relevance? Okay, that's an interesting biological observation. And so what we really wanted to do was take a tissue, add HIV proteins, and then add HPV particles, and see if that actually translated into more HPV getting in, right? Because that's our hypothesis. So the way we do that is we make virus-like particles, and we put a plasmid inside them. Instead of the HPV genome, it's a plasmid that expresses red fluorescent protein. So when the particles get into an epithelial cell, they uncoat, the plasmid is expressed, makes red fluorescent protein, and you can pick up a red signal. So if you see a red signal in a cell, that means that HPV has gotten in, or virus-like particles. So this is actually what happens. Here's a control, and here's the basement membrane. And then if you add TAT, one of those proteins, to the tissue, see all this red signal down there at the bottom? That's HPV getting in. You don't see that in the healthy tissue. So what it all adds up to is that we think that when people have HIV, even if they have well-controlled HIV viral loads, there's a lot of tat sitting around in the skin and GP120, and these things are basically making the epithelium